Biology revision video on synapses, and this is an update I'm making at the start of 2024 just because I wasn't quite happy with some of the stuff to do with excitatory and inhibitory synapses on the original video. From GCSE, the synapse is the part where two neurons has a gap between them called the synaptic cleft. It's about 20 nanometers in distance, which is something you need to know for A level. Action potentials can't jump across the gap, so we need a neurotransmitter as a result. And it jumps from an electrical impulse in the presynaptic membrane to a chemical impulse, and then to an electrical impulse generated in the postsynaptic membrane. We focus on cholinergic synapses where acetylcholine is the neurotransmitter. Here's the basic sort of structure that we need to be able to sort of talk through what is happening here. So you can see key parts of the structure are that there are calcium ion channels which open when the action potential arises. There are receptors for the acetylcholine, and they are made of 5 polypeptides with 2 receptor sites, and basically they're like a channel protein, and the channel opens when acetylcholine attaches. There we go. Okay, and that allows sodium ions to then rush in, which causes the membrane to depolarize to become more positive on the inside, and that then triggers a new action potential if you reach the threshold value of minus 50 to create an action potential. Have a look at the events then. So if we start off, the first thing that happens is the action potential arrives at the synaptic knob in the presynaptic membrane. So that's your first stage here. This then causes calcium ion channels to open, and the calcium ion channels then cause calcium ions to influx in. This then causes vesicles to do exocytosis. That's supposed to be a number 3, sorry, it's not very good. And those vesicles then fuse with the presynaptic membrane, releasing the acetylcholine across the synaptic cleft as a result. Okay, so I suppose that's number 3 if I was doing it properly. Okay, the acetylcholine molecules then diffuse across, and they attach to the receptors, which we saw in the previous slide, on the sodium ion channels. That causes the sodium ion channels then to open, and that causes an action potential because of depolarization if they cross that threshold potential of minus 50 across in the new neuron. Okay. Then in detail, well, the sodium, the action potential arrives in the presynaptic membrane, causes sodium channels to open and ions to rush in. This also causes calcium channels to open and calcium ions to rush in to the presynaptic neuron. This causes the acetylcholine vesicles to react to the influx of calcium ions by moving to the presynaptic membrane. Sorry, I've got to change that to 2+, it's not good. And that fuses it, emptying the contents into the cleft. And each action potential causes just a few vesicles to fuse, each containing up to 10,000 molecules of acetylcholine. We look at this in stages again, just different way, trying to give you as many different ways as we possibly can. The action potential arrives, causing calcium ions to open and flow in. The acetylcholine vesicles fuse and do exocytosis, releasing acetylcholine molecules into the synaptic cleft as a result. The acetylcholine molecules attach to receptors, opening sodium channels, but also potassium channels, but the sodium is the main one. And that causes the sodium ions to diffuse in faster. Some potassium ions do diffuse out, and an action potential is created in the postsynaptic membrane. To close the gates, we need to break down the acetylcholine to close the sodium and potassium channels. So therefore, we use an enzyme called acetylcholine esterase, and acetylcholine esterase is used to break down acetylcholine into ethanoic acid and choline, which is recycled and taken up using energy back into vesicles in the presynaptic There are different features of a synapse which sort of link to the point of them as well. They are unidirectional, so you only have receptors on the postsynaptic membrane, so it can only move from presynaptic to postsynaptic, and they are involved in something called summation. So there are two types of summation where low frequency action potentials don't always release enough concentration of acetylcholine or neurotransmitter to cause a new impulse, but it can build up either over time or from multiple neurons to trigger a new action potential. So there is one called temporal summation, where you might release a very small amount of acetylcholine into the gap at first, not enough to trigger one action potential in the new neuron, but if several of these action potentials arrive, that neurotransmitter builds up and it exceeds a threshold value to trigger the new action potential in the postsynaptic neuron. If there is something called spatial summation, so imagine neurons from your legs talking to your spine, lots of different action potentials from different neurons might come together, and together they combine to make a big enough impulse to go into the new neuron as a result of crossing a threshold potential. So this way you can have lots of impulses coming from lots of different parts of your body communicating and being trigger a new action potential in the next membrane. Some synapses are inhibitory synapses and actually reduce the likelihood of a new action potential being fired in the postsynaptic neuron. And they release a neurotransmitter called GABA. And the presynaptic neuron releases this neurotransmitter, which then binds to chlorine protein channels on the postsynaptic membrane rather than sodium channels. This then opens, allowing negative chlorine ions to enter the channel. To enter the membrane via facilitated diffusion. It can also trigger potassium channels to open, leaving the postsynaptic knob, causing a drop in the charge. The charge might drop even further away from a threshold potential. This means that you would have an overall negative even more negative resting potential, and you would need a lot of sodium influx to generate a new action potential as a result, because you get hyperpolarization, and therefore it inhibits the impact of the synapses similar areas. So you might get excitatory synapses near inhibitory synapses, and it is the balance of the two which decides whether a new impulse is generated. Synapse has lots of important roles. It makes sure that the impulse only flows in one direction because you only have receptors on the postsynaptic membrane. It allows integration, so you have lots of synapses might integrate towards one main synapse to respond one main postsynaptic membrane, and therefore that might bring in lots of information from lots of areas. We know the brain is full of synapses, and we know memory is about synaptic pathways and neuron pathways of different types. And summation is important because lots can come together to cause a receptor response in the postsynaptic membrane. And inhibitory synapses can obviously reduce the amount of unimportant messages that we process. And that allows us to filter out unnecessary things. It's a process called habituation. You need to be aware of certain drugs and the effects of certain drugs on synapses. This likely is to be given to you and you have to probably apply the information, but let's just have a look at a couple of drugs. So neonicotinoids are used in insecticides, so more likely in an exam you'd be told it's an insecticide that causes paralysis. They're not highly toxic to humans because we have less cholinergic synapses and binds less strongly to the receptors in mammals, but basically what they do, as you can see here, is normally the acetylcholine comes across, acetylcholine esterase breaks down the acetylcholine and it gets recycled. But what happens here is that the acetylcholine esterase cannot break down nicotinamides, and that leads to a blockage of the receptors. So the acetylcholine esterase can't block this, it blocks the receptors, and therefore you get paralysis as a result. dopamine, which is a stimulant drug, impacts synapses which use dopamine as a neurotransmitter, and basically it binds and blocks the dopamine reuptake transformers, so it stops it from going back into the presynaptic neuron, and therefore dopamine builds up in the gap and it continues to give an exciting feeling. Basically, all drugs are likely to affect the synaptic transmission. Nicotine is the same shape as acetylcholine, so it makes you that feeling of feeling good. It fits into the receptor protein, but it doesn't break down easily, and therefore it remains in a longer period of time. Large doses of nicotine can actually be Botox, which is actually a bacterial toxin, can cause a lot of illnesses, but we can use it to prevent muscle contractions. So some people use it to try and look young effectively. And it prevents the release of acetylcholine from the presynaptic membrane, so it prevents response. So it can be a deadly poison. It can also be used to relax muscles, and therefore can be used for cosmetics. Exams, it's unlikely you'll be asked to give specific examples. More likely they'll ask you to explain the impacts of drugs. So most drugs basically affect the impulse across the synapses, and they'll either stimulate or they will inhibit a system. So excitatory drugs are ones that might stimulate the release of neurotransmitters, which can increase brain activity, or you can have inhibitory drugs, which might block the receptors, and therefore it might stop the normal action of the neurotransmitter. Also, you could obviously have ones which prevent the breakdown of the neurotransmitter, which means that the receptors constantly are being exposed to the neurotransmitter.